So without further ado, Xingbo, you can share screen and we are happy to hear your lecture.
Yeah, sure. So let me share my screen now. All right, can everybody see it? Can start from beginning. Is it playing? Okay, good. All right, so I'm very happy to be back to continue with our lectures. So yesterday, for lecture 1, I talked about active matter theory and its application on the cytoskeleton self-organization using case studies of actual mouse networks as well as spindles. So I think the first lecture, in that case, is quite theoretical. It's a bit technical, but the idea is to give you this framework of active matter theory that can be used to study collective behaviors, pattern formation, and self-organization. And all those self-organization processes, they require energy. They are sustained by energy flows. And active matter theory is also a framework that takes into that consideration where energy are being dissipated at the local scale, in this case for cytoskeleton, at the molecular scales. But however, I would say this kind of incorporation of energy flows in active matter theory is still an open question because it was kind of implicit in the theory. And so therefore, I'm kind of proposing this framework in this winter school, which focuses on bioenergetics, also to kind of as a motivation for us to think about how to incorporate energetics into active matter theory. But first, we need to be able to measure energy flows, right? For example, to be able to do this kind of incorporation. And if we're looking at cells, if we want to study the self-organization of cytoskeleton, we want to be able to measure energy flows with very high spatial-temporal resolution, right? Single cell or even subcellular level resolution. So that's my topic for today's lecture, which is I'm going to introduce techniques, experimental techniques, to measure the spatial-temporal metabolism in cells with single cell and even subcellular resolution, to sort of provide the experimental tools for us to do those measurements and help us think about the subsequent study and modeling. Okay, so I basically ended on this note yesterday, like what is missing in active matter theory? Well, there can be many things that could be missing, but for the sake of this, winter school, I think also, which is a very important topic, what could be missing is this explicit accounting of energy flows in the active matter theory. If we're talking about in vivo or in cells, it's the metabolism, right? Cell metabolism, cell energetics, that is actually missing in this active matter theory framework. For example, if we look at a cell, which I'm showing a schematic of an oocyte or an egg, which actually going to be, a very important organisms for throughout these talks. This is also the systems we are studying. And where you will have a steady state spindle arrested in this metaphase 2 phase, waiting for fertilization. And this spindle, again, is active structure, right? It has this energy, and energy for oocytes is primarily provided by mitochondria, right? A process through mitochondrial respiration that, for example, produce ATP. ATP would be utilized by the spindle, forming this cycle. So, and this not only happens at this single cell level, it happens on, on the multicellular level, right? If you fertilize this egg, it starts to divide and form early embryo, and then made of many different cells. And, then the cells are moving and dividing. You're involved in this process of morphogenesis. And that also takes energy. And the energy can be supplied, again, by mitochondrial respiration or by glycolysis, and in the form of ATP. And that powers the cell proliferation and cell migration. And that together, interacting with all the genetic and mechanical program, that drives morphogenesis. So there have been extensive studies on the dynamics and mechanics of self-organization, right? Including the cell mechanics including all the genetic programs, for example. However, much less is known about the energetics, right? Which is very fundamental, but even questions like what are the energetic costs of those different cellular processes? How much energy does it take to build and maintain a spindle? How much energy does it take for a cell to divide, to migrate? We don't know this, right? We don't even know a clear energy budget of a cell. I'm sure this will be a recurring theme throughout this school, and there are many other speakers who also kind of provide different perspectives on this topic. But I think this could be one of the very, important questions for us to start addressing. And I'm going to propose some kind of ways to start, going to address those questions. And I think you guys will also have a chance to think about it during the talk and during tutorial sessions, for example. And then the other questions are, what are the energetic constraints, right? Either imposed by thermodynamic laws, for example, physical constraints, or constraints from material properties, because this, cells are made of these molecular machines, and there could be intrinsic constraints imposed by the material properties. And also, what are the impact of energetic perturbations, right? Which also, even from an applied perspective, is significant because there are metabolic diseases, right? If there are problems with these metabolic pathways or in the energy flows, that could have subsequent consequences in the cell physiology, which involve many diseases, including infertility, including cancer, neurodegenerative diseases. So those are all worth thinking about. So first, in this talk, I'm not going to go into details about the metabolism. I think many other speakers will, do the job. I'm just going to give you a very coarse grain view, right? The coarse grain, this word keeps coming up, right? Think about it as a kind of top-down perspective where you think things in terms of modules. And I'm only going to give you this kind of overview, and so that provide you necessary information that we can to kind of motivate our measurements. So overall metabolism, I view metabolism as basically you have a cell, which you can consider as a black box, and then you have nutrients from the environment or supplies that basically is being uptaken by the cell, and then you just secrete waste. If you just look at the cell at the black box, this is what's happening. You don't know what's going inside. And this is basically a global energy flow. But if you look inside, there are many different cycles. It's basically this kind of global flow is driving those cycles. One of the very important cycles is the ATP-ATP cycle, right? So there can be glycolysis happening in the cytoplasm of the cell that is basically taking glucose and breaking it down eventually to CO2, but that would use the mitochondria, as I will discuss, and then you would produce ATP to harness the free energy from this reaction and to make ATP. And then there's mitochondrial respiration. So this organelle, or the powerhouse of the cell, as you guys must have heard of this in your cell biology textbooks, where one of the critical functions of this organelle is that it also breaks down nutrients. In this case, for example, it can break down the nutrients that can break down the product from glycolysis. Like one glucose can produce two pyruvates, and this pyruvate will be a substrate that can go into mitochondria and will be eventually oxidized into carbon dioxide, and that would produce ATP. There's another very important ATP production pathways. So on the other hand, so this comes a bit earlier than expected, but this other part of the cycle will be all the ATP consuming processes. So you have these two ATP production processes, and then you have a lot of ATP consumers, right? Including protein synthesis, cytoskeletal assembly, ion pumpings, among many others that are consuming this ATP. So that forms a loop, right? You have production and consumption, and basically cell metabolism in a very simplified view are composed all of those cycles inside. Okay, so sorry, my slide is somehow playing automatically again, like last time. Oh, sorry. Let me. Can you guys see it again? Can you guys see the slide? Good. Okay, so I want to make sure it goes on command. So okay, let's try it again. So and during these cycles, right, there are those fluxes, right? So there are concentrations of ATP, ADP. And also there are those fluxes, which characterizes how fast you are, for example, making ATP or consuming ATP. And these are the turnover rates of the molecules. And they characterize the metabolic activities. So that's kind of an overview of cell metabolism. And then, so here I'm showing you a few real case studies sort of to motivate this connection between cell metabolism and a lot of the cell processes. For example, here again, you are looking at a oocyte, in this case, a mouse oocyte, which is arrested at metaphase 2, where you have this nice spindle at the near the cortex, waiting for again fertilization. And you also can have mitochondria around them, so the mitochondria everywhere. And now, so to show you there is a connection between metabolism and spindle self-organization, we did this experiment of drop and recover oxygen, which I also showed towards the end of my last lecture, where you can see that if I drop the oxygen, which is required for respiration, you can see the spindle starts to respond. In this case, it shrinks, becomes smaller, but in a reversible way, right? So when you drop oxygen, very slowly here, the movie is about 2 hours. Now, disassembles, probably disassembles, and then if you recover oxygen, it comes back. So there's definitely a crosstalk, right, between the metabolism and all those energy users in this case, right? I have to say that this is ongoing work, and we are still trying to strive to figure out what is the actual connection here. It could be maybe as a hypothesis as ATP, where when you drop oxygen, right, and there's mitochondrial respiration is inhibited, and where I just showed they are primarily ATP producer in the cell, and your ATP level could decrease, and maybe then the spindle is impacted through that. But that's not the only hypothesis, many other factors. But to disentangle and to figure out the mechanisms, we need to be able to measure those quantities, right? We need to be able, for example, to measure ATP, if that's a hypothesis, in the living cells, during this experiment, and potentially with even subcellular resolution, because we don't know if ATP levels are homogeneous or not. Yes, Yash?
Question. So I'm looking at the right movie, and as you see the spindle shrinking, if you look at the neighboring dots, and lots of things floating around, they all seem to slow down.
Yes,
is it really
good
like
observation.
So it's like a temperature has gone down, a local temperature?
Yeah, super good observation. Yes, actually, they do kind of, so they do, right, starts to kind of freeze. We don't know what's going on there, but, you know, there are also work, I think in this case, in bacteria, I might quote that, where people also start to de-energize the cell by also inhibiting mitochondrial metabolism. And they also showed that the sort of diffusion of some of the, different particles inside of the cytoplasm got slowed down, and they classify that transition to sort of a glassy state, right? So I don't know if this is related. You mentioned temperature in this case. Of course, these are all very active systems, right? So I think the active matter there, this kind of thermal diffusion of that is quite negligible. So a lot of those motions are presumably driven by active processes. Let's say, for example, they're probably embedded in the cytoskeletal network, and the cytoskeletal network's dynamics is powered by ATP, and they are like sliding and doing all those things, as I talked about last time. Maybe those things got slowed down. So that could be a hypothesis, but it could be maybe other possibilities there. So I think they're kind of a related question there that we have to address by being able to first quantify them, maybe tracking them, and then being able to characterize what's going on in the metabolism.
Thanks.
I had a question. So regarding the depletion of oxygen in the cells, is there any influence of hypoxia or hypoxia-inducing factor, the HIF1 factor protein, which probably might be related to the action of spindle fibers? And is there anything known about that?
Yeah, very good question. So HIF factor, yes. I want to emphasize a bit. So HIF factor, for people who don't know, is a transcription factor, right? I think that is also related in signaling. And when the oxygen is low, that HIF can be activated and also translocated into nucleus and upregulate transcription, etc. Here, a lot of the perturbations I will be talking about today, including this one, it's very fast. right? It's a very acute perturbation. In this case, okay, this particular movie is slightly slow, but it's still like 1 to 2 hours, right? The drop is about 1 hour. I can do it faster. I've done it faster, where I can literally drop within 5 minutes, and they're just gonna respond within that time scale, right? 5 to 10 minutes. And I suspect during this time scale, it's kind of HEAF, response is probably not relevant. Another thing to note is in this particular case, the transcription should not be active, because first, there's no nucleus already broken down. You're actually at metaphase. So you have actually chromosomes already compacted right here in the middle, right? So there's actually no transcription in that case. So this is kind of, in that case, it's quite a simple system, right? So you probably don't think about those transcription and out of those things, you think about things from a biochemical perspective. And also because everything happens pretty fast, the perturbations are done very acutely. Okay, right? So I think it's a very good question. I think this also sort of sets the stage of what I'm about to discuss, right? I think a lot of I'm going to talk about is about, in this energetics, it's about biochemistry, this kind of kinetics. But I think there's another other side of the story, which is this type of transcriptional regulation and everything, which could totally be a very, very big. I think it's probably better, much more well studied than what I'm going to be talking about in this kind of lectures. But I think this is the things that we also need to, take into account and study more in detail.
I have a small question.
Yeah.
You said that the active processes, all the active processes in the cell would get affected when you decrease the oxygen, because the mitochondria is not producing energy anymore. But what about the passive processes which just rely on the thermal energy? Since the mitochondria is not producing enough energy, so it's not convecting enough energy in the cytoplasm. So do you also expect the cell to, do you expect some temperature change as well?
Oh, temperature change. Okay, you bring another very interesting question. So, okay, I would say, okay, so there is, okay, I can probably, okay, so I would say, then you can estimate in this case, like, for example, you can ask this question: would the mitochondrial respiration, would this kind of process being able to, let's say, impact the temperature of the cell? Right? Or let's say if we can even generate a temperature gradient. Actually, people ask this kind of question, and there's actually one work which is also, stirred up a lot of debate, is saying mitochondria is pretty hot. So they claim that mitochondria is can be up to 50 degrees centigrade, or in other words, like about 10 degrees hotter than the cytoplasm, the rest of the cell, right? So this is like a local furnace. And if you inhibit the mitochondrial respiration, you can decrease that temperature difference. But also there are other works showing that from kind of physical type of, okay, so that work is the conclusion drawn from some biosensors, some, well, I would say not, so from some sensors, temperature sensors, that they can embed in, I think maybe like some nanosensors that can put into the cell, and they calibrate it and they make that measurements. But then also people have done back the envelope calculations and knowing the respiration rate, right, of the mitochondria, and then argue that it's probably unlikely to generate such a big gradient. So therefore, I think it's still up to debate, right? Would this kind of mitochondrial respiration change temperature of the cell or even gradient? So I would say it's a very good question, and we should keep thinking about it.
And I have another question, which is a more practical one. So could you control for that in your experiments, since you use a pressure chamber, and when you modify the concentration of oxygen, I assume that there would be a pressure change that would also induce a temperature change in the system, right?
Yeah. So okay, I don't know if the pressure change will change the temperature, but I do control for the environmental temperature. That's a very, very important factor, especially for those very sensitive mammalian oocyte cells. We always, for example, like the optimum temperature, right, you do is like 37 degrees, and you control that in the box, in the chamber. So that has to be very well controlled. And we can also change the temperature. So this is some information here. We did change the temperature. So I did do experiments of changing temperature, actually all the way from about 20 degrees, 20 degrees C, all the way to 40 degrees C. And I definitely see very strong change in the respiratory rate of the mitochondria of the cell. I did measure that directly. And so that's a very, very interesting observation. And actually in my lab, we're pursuing that. We're trying to understand how does mitochondrial respiration rate change, in response to temperature change, and trying to develop a thermodynamic model for that.
Okay.
So I would say stay tuned.
Thank you.
Yeah, good. All right, cool. Thanks a lot for those great questions. And then, here I would like to give you again, mostly especially students, sort of just again a movie, show you the very fundamental, energy production pathways in the mitochondria. So let's go with the movie.
Our body is made up of trillions of cells. They all require
Can you
energy
hear
to
anything?
function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of two membranes, an outer membrane, separating it from the cytosol, and an inner membrane, surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
Okay, so I think this is a very elegant and straightforward explanation of this electron transport chain, which actually it would be which is at the kind of at the center of this mitochondrial energy metabolism or oxidative phosphorylation, as in the movie has explained. So here is kind of a recap. Again, I'm taking things simple, right? So what basically this mitochondrial metabolism is doing, as shown in the movie, is that it takes in nutrients, right? For example, pyruvate in this case, and then they use oxygen to oxidize the nutrients essentially, and then in this process it transduces the free energy through a few cycles inside of the mitochondria, and essentially the free energy is used to make ATP, and essentially ATP is being used by the other cellular processes. So if we look inside of the mitochondria, as shown in the movie, you basically have this 3 cycles. So I want to simplify things for you. So I just want you to remember, if you think about mitochondrial energy metabolism, it's 3 cycles here. In this case, what I mean is that there's a NADH cycle, right? Which is NADH is this electron carrier, as we show in the movie, right? This kind of enters into the first complex across the electron transport chain. And that is the electron carrier. So this is kind of a very important molecule, right? People know a lot about ATP, but sorry, it bothers me that the playing is automatic for some reason. Maybe let me take one little or two. Okay, let me try to start this again. Okay. All right. So now, so like I said, NADH is also very, very important molecule. So that's kind of the first cycle, right? Where you will use the free energy from the nutrients to reduce NADH. NADH is a redox pair. So things happen in pairs, right? Because there are cycles, right? So inside of the cell, so they always come in pairs. So in this case, that NADH can be in a reduced form or in an oxidized form. The reduced one is called NADH, oxidized one is NAD+. And then, so in this kind of the nutrients, we'll go through a few cycles inside the mitochondria to make NADH from NAD. And the NADH now would participate in the electron transport chain, where it would donate its electron to oxygen. And so oxygen is the electron acceptor. And then that free energy, right, harnessed by this electron transport through the electron transport chain would be used to pump protons across the mitochondrial membrane. And then now the free energy is stored in this proton gradient, right? And this proton gradient will be used to power the ATP synthesis through this particular molecular machine, which is called ATP synthase. Of course, this process is not perfect. There's always some leakage, and that is the flow of protons across the mitochondrial inner membrane that is not used to make ATP, right? It could go through some other channels or some just basal leak through the membrane. And then now the free energy now is transduced into ATP now, right? So it basically kicks ATP out of equilibrium, right? Because at equilibrium, ATP will be at a particular ratio. Now with this proton motive force, it's kicked out of equilibrium. So you have more free energy now. And now this free energy can be used through this ATP hydrolysis and to power the other cellular processes, right? So in other words, this is basically a free energy transduction, right? The energies are driven, all those pairs out of equilibrium, one at a time, and to turn by turning those cycles. And so there's a coupled 3 cycles. Okay, so that's kind of mitochondrial metabolism in a nutshell. And then to measure, mitochondrial metabolism, you just need to measure the quantities related to those 3 cycles, right? So one of them is this NADH, NAD redox cycle, and then you can just measure NADH or NAD levels, for example, right? You can measure the concentrations, you can measure their ratios, right? That also signifies how far away they are from equilibrium. You can measure, of course, you can measure oxygen, right? Because that's something comes from outside, right? All the other stuff is basically inside, right? They're just inside at their respective steady state. For example, in this case, for oocytes, oxygen is what comes from outside. So you can literally measure the oxygen uptake rate, right? Of course, you can also measure the nutrient uptake rate, also come from outside. And then the second cycle, you can measure this proton gradient, right? Like across the mitochondrial inner membrane. And that is the proton motive force, okay? And then, of course, you can measure ATP concentrations or their ratios as well, right? So those are the things that in this talk, I will be introducing techniques to basically measure them, right? And so that you know what the tools are available now. But I would say, right, new tools are being developed. And I think, if people develop new techniques for those, it's always gonna boost the, field forward. So how do you measure NADH, right? So let's say concentrations. Okay, here's a good news that NADH is autofluorescent. I think this is a very, very cool fact that you don't need a label, right? So that means what is autofluorescence? Autofluorescence means you don't need any external label, that if you just shine light, excitation light, according to the excitation spectrum of this molecule, in this case, it's about, this is the excitation spectrum of this molecule. Any fluorophores have such a signature spectrum. And the peak is about 375 nanometer. So if you tune your light, 375 nanometer, you excite this molecule, right? When you're excited, what happens is that this molecule is going to be excited, right? Go to excited state, and then it will decay from that steady excited state, and then we emit a photon, right? So, and the photon will be a little bit redshifted in this case, and then the emission would peak at another wavelength, which is about 460 nanometers. So now you just put a detector and put a filter that filters out most of the other light, but focus on the 460, and then you will be able to see this molecule, right? And people have calibrated and tested in cells, and if you do this excitation and emission, you will basically be looking at NADH, which is predominantly enriched molecule. And also another one is actually I did not show is NADPH, which is actually another electron carrier that is involved more in biosynthesis, right? For example, more outside of mitochondria. But here, NADH is very much enriched mitochondria, and if you just tune your light and into this excitation and emission, according to the spectrums, you see them. Okay, so therefore, it's a very general method, no label needed, can image any biological sample with the right excitation emission wavelength using a fluorescence microscope. Okay, so that's very good. So here, just to show you that we did it, for example, for mouse oocytes, for human tissue culture cells, for yeast, right? You just use the exact same excitation emission, you'll be able to see all the NADH inside the cells. Is that a question? I heard some.
Yeah. I just had a small doubt from the previous slide. So yeah, as you were mentioning, there is like in the NADH autofluorescence, there is also contribution from NADPH pools. So since their ratios inside the cell and also in different compartments are largely different, so like, are there any methods to segregate these two? Because I feel that there might be, like, it can skew the calculations a lot if we only rely on fluorescence.
For sure, exactly. So that is a challenge, right? So if you just look at fluorescence intensity, you cannot separate them because their spectrums are identical, right? Of course, there are cases, for example, in the mouse oocytes, if you do a mass spec, right? So mass spectroscopy, you can see that NADPH, now you can distinguish the different mass. NADPH is much, much less, right, than NADH, order of magnitude almost less. And then you can basically safely assume that most of the signal you see is NADH. That's just a minor contribution. But of course, for a lot of growing cells, that might not be the case. For that, I think if you want to account for that, you can there are biosensors now. I think that's why people develop biosensors that can specific to NADH or NADPH. Then you can use those biosensors, they're protein-based biosensors that can press them in the cell, and then you can calibrate them, and then you can basically disentangle these two. There's also kind of quite recent work which people use what is called fluorescence lifetime image microscope. Right? So fluorescence, right? So here I'm just talking about like you can count photons, right? You can look at how bright they are. But actually, like I said, the fluorescence happens when you excite a molecule and then it takes some time to decay. So the time it takes to decay, that's called fluorescence lifetime. There are actually very accurate, instrument to measure that, which actually I will be talking a lot in my next lecture. And this technique will be able to, according to this one study, that be able to distinguish NADH from NADPH because they have different fluorescence lifetimes, especially when they are bound to different molecules. So the caveat is that I think this method is not being able to distinguish the free NADH and NADPH. They can distinguish the enzyme associated NADH and NADPH. So those are kind of the state of the art.
Yeah, thanks. I was actually going to ask that. So like, can you differentiate between the protein bound and the free NAD pools? But I understand.
Yeah, stay tuned for the third lecture.
Thank you.
That's what it's all about. Okay, but the question is, the answer is yes. Okay, all right. So and then so now this you can basically see that, and also you can see that there are brighter pixels, and those are mitochondrial pixels because NADH is very much enriched in mitochondria. So in that case, NADPH is less of a problem if you segment, right? The mitochondrial pixels, which I will talk a lot about in my next lecture. Okay, so now, okay, so here actually is a little bit of already a kind of a foreshadowing here, where so for example, I like mouse oocytes a lot, right? Because they are at a steady state, and right, so meaning they have a kind of constant metabolic activities because their cell cycle arrested, right? As there's not much of even transcription happening, a perfect system to study this type of energetics, right? From a kinetic and the biochemistry perspective. So I imaged this, and then, right, so those brighter pixels are actually mitochondrial pixels. And how do we know that? You label mitochondria with a mito tracker and actually co-localize. So that's how we validated it. And even just based on NADH contrast, you'll be able to segment very nicely as we've been benchmarked with the ground truth. So that's what I mean. And if you want to study mitochondria, you want to segment mitochondria from the red cell. Okay, you can do that with NADH. Okay, so here I'm also showing that NADH it's a natural biomarker. It's very sensitive to oxygen perturbations, right? So I showed oxygen perturbations that change in the spindle. I think a lot of people already asked me, like, how does that change mitochondria? So here's the answer, right? So if I drop oxygen, this is basically the experiments I was doing, similar to the ones I did with spindle, that again, the first cycle now, right? This oxygen will be needed to oxidize NADH. So I drop oxygen very slowly and I recover it like so. And here is a movie showing how NADH responds. All right, this is a recovery, so it's reversible, so it's playing, and now I'm dropping oxygen. I'm recovering it. Okay, so I can quantify this by looking at the mitochondrial pixels again, right? So I can segment the mitochondrial, integrate its average intensity, integrate its intensity, or calculate its averaged intensity in all the mitochondrial pixels, and they increase as oxygen is depleted, which is as expected, because then you cannot oxidize NADH, so that accumulates, right? And NAD plus is not fluorescent, so that's why you see this. Okay, so next cycle is proton.
Xingbo, can you play the movie again?
Okay.
Give me a sense of the length and time scales.
Yeah, the time scale, as you see, is a 1 hour here, right? So the drop is about 30 minutes. I just do that because I just drop it with that time scale, right? You could drop it much faster, the response will be much faster, but I choose to do that. And then the length scale of the whole cell, the diameter is about 80 microns. Sorry, I did not include the error bar, the scale bar. So 80 microns across. Pretty big
signal
cell.
that we are seeing is coming from within the mitochondria. Is that right?
Can you say again?
So the signal that we are seeing, the white spots in the movie,
Yeah.
so that's coming from within the mitochondria.
Yeah, those are mitochondria. Yeah. So if you put a mitochondrial tracker, and they're basically going to colocalize with those brighter pixels. Yeah. So that's what's being quantified here. I just integrated those brighter pixels.
So I had a question on this slide. So
Yeah.
you say that when the depletion of the oxygen is being recorded as the intensity of NADH. So you're saying that when oxygen decreases, NADH is accumulating, so we get to see the intensity. So aren't there other mechanisms apart from oxygen that might also lead to the accumulation of NADH? And hence, we probably cannot directly correlate the two, that only the depletion of the oxygen is causing this accumulation of NADH and not other mechanisms inside the cell.
Yeah, very, very good question. So as you can see, even you just look at this cycle here, right? Even just look at this, this cycle on the left. So there are two ways to increase NADH, right? One is you sort of decrease this oxidation of NADH. So this right arrow, if this is being inhibited, for example, decreasing oxygen is one way to decrease the oxidation of NADH. And then NADH would accumulate if this, for example, if this left sort of cycle is not changed, right? But there's this left cycle, right? Left arrow, which is the reduction of NADH from NAD, which is the production of NADH. So another way to increase NADH is if you just, let's say, maybe the Krebs cycle runs hotter, right? For example, if you have more nutrients, right? And that drives the production of NADH more. And then you would accumulate NADH as well. So you can either increase NADH by increase the production or by decrease its consumption, right? That's a cycle. So therefore, okay, you can say when I drop oxygen, maybe the other possibility is somehow if I drop oxygen and then this cycle increases, right? It's I would say, I think we can rule that out because your system is at a steady state. That's why I drop my oxygen very slowly, right? All the chemical reactions very, very fast. So therefore, these two arrows will always have to balance each other. Okay, so quasi-static. So when I drop oxygen, I actually measured the NADH consumption rate, and that decreases. And that also means this left arrow is also decreases. So in this particular case, I think it should be attributed to the decrease of consumption rate, right? But also there are other ways to decrease the consumption rate. There's a lot of enzymes here. You can inhibit those enzymes. We'll see similar phenotypes.
Right. So I was just wondering if you could somehow halt the process, like as a control experiment. I was thinking if you could halt the process of, conversion of pyruvate back to CO2, and hence the increase of NADH is absolutely not by that mechanism. And any other mechanisms in the cell that might also lead to accumulation of NADH, if we halt those, and then we can say that, yes, it is because of depletion of oxygen that this. So are there control experiments for the data, or this was the first?
That's a very good question. I think, yeah, that's a very good idea. I think we can hold it. We can stop it at the intermediate steps. But if you're talking about if I'm going to hold it and then perturb pyruvate, is that what you suggested? One of the possibilities?
Yeah. So anyhow, like in that entire mechanism by which pyruvate is converted to CO2, and that leads to accumulation of NADH, right? So any mechanisms, whatever can lead to accumulation of NADH, if they are simultaneously halted, and then the decrease of oxygen is causing this, then it might mean oxygen is directly causing the accumulation of NADH. Otherwise, there might be other mechanisms which is caused due to depletion of oxygen, and that is indirectly causing the accumulation of NADH. Hence, we might not be able to directly correlate the two. So that's what I was
those are good points. I think this exactly. So I would say those are very good ideas. There could be some challenges in there because, like I said, those, for example, if you were, but I would say like it's totally worthwhile doing, and I think it's something one should try. I'm just posting some things to think about, right? For example, like these two cycles, right? The reduction and oxidation are always balanced. In this case, if you were to perturb one, you are definitely going to impact the other. So that's kind of poses a bit of a challenge there. But maybe there'll be some other, let's say maybe some signaling or some other redox control pathways where they are. Maybe those are kind of affecting NADH, and those can probably be simultaneously suppressed and see if there's any changes or not. So those are good ideas. Okay, good. So so that's NADH, and those are very good questions. All right, and then the next one is the proton motive force, right? So the proton gradient. How do we measure that? So now, for this, since for protons, they're not autofluorescent anymore, right? So you can just image them directly. Therefore, you need to have. So in this case, right, where I'm focusing all on kind of microscopy techniques, right? Because that's the technique that will be able to give you the optical resolution, right? So another way to visualize some molecules, which by themselves are not fluorescent, is that you take advantage of some other fluorescent molecules, right? So in this case, there's this molecule called, it is commercially available, which actually being extensively used in metabolism research, which is one of the very easy thing to do. If you want to do metabolic research, of course, you're going to do NADH. Next easiest thing to do is probably this dye, which is a TMRM, which is kind of a membrane potential sensitive dye. The dye itself is charged, so therefore it will co-localize into mitochondria, which is more polarized, right? Have a larger membrane potential. In which case, and actually it's actually quantitative, but of course, to make it quantitative, you really need a lot of calibration. But this dye follows this type of, if you can call it a nurse equation type of kinetics, where their accumulation inside and outside of a charged membrane will give you the actual membrane potential, right? Following this equation, if you can measure the inside accumulation of this molecule, because, for example, because of this proton pump that that would establish a proton motive force and therefore a membrane potential, therefore potential gradients, and then the dyes would accumulate based on this kind of gradients differentially into these two compartments, and then the in and out would have different accumulation of the dye because they have different potentials, electric potentials, and then this equation gives you the membrane potential difference across the membrane. So if it is depolarized, then you basically, if it's completely polarized, and then you would have no, this preferred accumulation in one compartment versus the other, you have a homogeneous accumulation. So that's zero membrane potential. Look from this equation, it's log 1. Okay, so for that, you can actually see, people usually use this in a more qualitative sense. Okay, because to be quantitative, you really had to calibrate, the concentrations very, very carefully, like you have literally measure the concentrations exquisitely accurate. And it's very much more difficult to measure concentrations than just to measure the intensity, which is, you know, in that case, more qualitative. So for that matter, you can just look at intensities, right? So if you put TMRM inside of this, in this case, neurons, and they're mitochondria, right, inside of the neurons, axons, in this case, it's all over the place. And then if it's a polarized mitochondria, then the dye would accumulate inside more into the mitochondrial matrix, and you have a higher intensity. And if you depolarize the mitochondria, and then the membrane potential collapses, and then the dye would not accumulate, right? If it's completely depolarized, the dye will be homogeneous everywhere. So therefore, you would see this decrease of the signal. So that's one way you can actually measure membrane potential. Okay, so for example, we also did this, use this dye in this mouse oocyte on the exact same experiments of oxygen, you know, drop. And if we do that, let's see, this is what happens. Okay, so pretty fast. Let's do it again. Oxygen drop, recovery. Okay, let me actually, I wonder if I can actually pause this somewhere. Okay, so this is starting to depolarize. This is like, okay, this is recovered, sorry. So this is the beginning, hyperpolarized mitochondria. This is starting to depolarize. This is like truly depolarized, right? So I really want to capture this depolarized state. So here, right? You can see that the contrast between the mitochondria and cytoplasm decreased, right? If it's completely polarized, it can become homogeneous, right? So, and it's reversible. So when you recover oxygen, then you kind of, I mean, the idea is, right, if you drop oxygen, you stop sort of pumping the protons, in this case, from the matrix to the intermembrane space, right? That's what I mean in and out. In this matrix, out is the intermembrane space. And then if you drop oxygen, because oxygen would be used for this electron transport chain to happen and for the proton pump to happen. If you drop oxygen, then the pump stops, right? And then that's why you collapse the membrane potential. So that's kind of consistent, okay? All right, so any questions regarding this? Yeah?
Or just one question out of curiosity. So how fast does this dye basically relocate? So what's the temporal resolution that you can obtain with these kinds of measurements?
Very, very cool. Exactly. It's a great question. I think this dye, based on our empirical results, is pretty fast. So look at this right here. The recovery is like within 1 minute, more or less, 1 to 2 minutes, right? So that's a very, very fast recovery. And then you can see that drop, the recovery is almost instantaneous. So if we did this with a confocal scanning microscope, right? So each image is takes 30 seconds to scan, and I'm doing continuously. So therefore, like one frame versus the other is probably like 30 seconds apart. So after the recovery, between this one and the next frame, it almost recovered. And again, it takes another 1 or 2 frames to completely recover. So I think the relocation of the of this dye is very, very fast in that case, like within a minute.
Thank you.
Hello. Yeah, I had one question regarding the like on the slide. So as you were dropping the oxygen levels,
yeah.
So before you start dropping it, there has to be some membrane potential that's present in the mitochondria. So as you're dropping the oxygen level, how exactly is it being dissipated?
How exactly it's been dissipated?
Yeah, because I would expect.
Exactly, can you elaborate a little bit more? Like, what do you mean? How exactly?
Yeah, because I would expect the oxygen to be important for its dissipation. If you are reducing the oxygen, then how is the how is the intensity going down? That would mean, like, what is the alternate route that the membrane potential can be dissipated through? Would I not expect a lowering in oxygen and a buildup of the membrane potential?
Okay. Yeah, see, so this leak, for example, right? So this is how it dissipates. There's leak, and also, of course, there's this dissipate through by making ATP, right? So if you want this oxygen, consumption is used to help pump the protons. I think there are some really good questions going on here. I think even including the questions about NADH. So this is where I think maybe the take-home message is think things in cycles, right? So for so for any kind of concentration to build up or dissipate, there's always two routes. One is you increase the production. The other is you increase, decrease the production. The other is you increase or decrease the dissipation or consumption, right? So in here, the oxygen is at the production side, if you want, right? So if you have more oxygen, then you actually can run the electron transport chain, and then you can actually run this arrow. That's directly coupled to oxygen. This arrow means you are pumping proton from inside of the mitochondria to outside. And when you do that, you build up the memory potential, right? So, and then of course, what goes out, have to, go back in, right? At steady state by definition, that therefore it has to go back in through either leak, right? There's some channels in the membrane that just can leak through, or they can just go through its ATP synthase, which is actually the key of making ATP, right? So that's how the memory potential is used to make ATP. So therefore, if you drop oxygen, then let's say you are not changing the leak and the ATP production, right? They don't see the oxygen, right? So then your kind of pumping starts to decrease and your dissipation keeps the same, stay the same. And then you have to reestablish your steady state. That means your memory potential has to collapse.
Okay, so the oxygen tension somehow has a feedback loop on the reducing equivalents that are being oxidized, because that's how you reduce the pumping, but you keep the dissipation constant.
Yeah. Or you can say the lack of feedback.
Okay. And is there any way to figure out if the rate of leaks change in these scenarios through the membrane?
Yeah. So very, very good question. So I think I might touch a little bit on the last, my third lecture. It's slightly difficult to measure a leak. A lot of those intrinsic fluxes, inside the cell is quite difficult to measure because, for example, it's hard, even if you can measure the dissipation of protons, it's hard to see if they are through the leak or through the ATP synthase. It's hard to distinguish, right? They both just like go through the membrane. So there are some ways where you can presumably, block one channel and see what happens to the other one. Right? You can block the ATP synthase and see what happens to the leak. And that's kind of some tricks I did to kind of disentangle this. But in terms of how this changed during oxygen drop, I think I have some very preliminary data, but I probably would not go into that in these lectures. But those are the kind of the ways you can probably help you. Let's keep that open question.
Okay.
Yeah. Cool. All right. So then let's go to the last cycle, right? Oh, yeah, Yash. One more question.
So I have a question.
Oh, yeah.
So I had the occasion to read some article about TMR and rhodamine derived probes.
Yeah.
And I was wondering, do you know how much is this relevant to learn about mitochondrial physiology? Because I saw that TMRM also onset depolarization. And so then thanks to those probes, you can learn about, for example, electric continuity of mitochondria and synchronicity. But then because you onset depolarization, then you don't know what is like the normal potential of the mitochondria.
You mean the TMRA would change the memory potential by themselves?
I saw that you can measure the potential because of the gradient of the probe inside and outside, but then you can see that the presence of like the TMR, the TMRM or rhodamine or whatever also onset depolarization in other like site of mitochondria when they are connected, luminally connected,
Okay, so I think if I understand correctly, you're asking there's probably some artifacts in there like from this dye. That's the interpretation of your question?
Sorry, what?
Did I interpret your question right? That you are saying there might be some artifacts from this dye?
Yeah, but also because the general question will be, is it okay to insert some charge probe into mitochondria because of oxygen reactive and something like that?
Yeah, it's a very good question. I would say, okay, personally, I did not know much about, it's totally possible, right? So I'm sure it's concentration dependent as well, right? So usually when you use these dyes, I think for any dyes, I would say there will be that problem, right? Even you label, let's say, spindle with thutupalli, right? If its concentration is too high, you actually would inhibit the process you're measuring themselves. That probably goes for any measurements. So for that, I think one thing you can do is like you can titrate the dye, right? Like you can go from a very low concentration and gradually increase it, and to see if your measurements are perturbed, right? From an empirical way, that's something one can always do. I think this maybe also is a way. Actually, I would just give it out there, experimental way to test those artifacts. Another way is, of course, you can benchmark it with some other technique, right? So there are first there are actually other ways, which is there's a patch clamp technique, right? So which is actually more, that's probably the most direct way to measure membrane potential, which you literally put the electrode into the cell. People use that in neuroscience a lot, where they just measure the membrane potential, the cytoplasmic membrane potential. It's much more difficult to do it for mitochondria, but I still know that there are people actually did that with isolated mitochondria. Then you can patch clamp that. So I would say another way is to patch clamp it and sort of to literally read off the direct actual membrane potential and to benchmark with this dye if you want to worry about artifacts. But other than that, I think, for example, experiments like this will show you that this dye would be good in this, at least give you this kind of qualitative trend of response.
And so would you have a solution when we are trying to figure out the gradient, the proton gradient, but in organs, for example, not in just cellular culture?
In the organ, you can also use this dye. For example, this is you can be scaled, right? People do that as well. People do that for organs, right? You can image the whole organ, and you can actually see the distribution of the membrane potential using this dye when you optimized it. I think that people do that all the time. So this is kind of can be used on subcellular, cellular, and tissue level.
Yeah, but then you will not be able to put a patch clamp on. No.
Yeah. Then in that case, another way you can do this, I think I probably can refer to one work where people actually calibrated this dye quantitatively by permeabilize. Okay, let's see. Did they not permeabilize itself, but actually by kind of inhibiting some of the ion pumps, and then sort of being able to titrate the extracellular, its potassium levels, and to actually biochemically clamp the membrane potentials, and then use this dye to measure that. And actually that responds very nicely, right? When you kind of titrate the potassium levels outside of the cell, and that actually changes your membrane potential also of mitochondria. And there they actually even produce this type of calibration curve of TMR like intensities and actual membrane potential in millivolts. So I would say that's probably the right way to use this dye, right? That can be done in your different organisms, but you just have to do that calibration.
Okay, thank you.
Yeah. Okay, so yeah.
It's a simple biology question actually. So these protons that you are talking about inside or in the intermembrane space, these are not free protons. They are bound to some molecule. I'm guessing water or something.
Good question. That I would say I don't quite know that. I don't know the answer to that question. I was assuming they were, I'm sure they can definitely interact with many other things, but yeah, I think, I don't know how much are free versus how much are like bound. And what's on your mind? For example, what would be the implications?
No, so what I was thinking is that, well, free proton is just it's a proton, so it's a very small thing. So if it were free or if it were bound to something very small like a water molecule, why can't it just diffuse through the membranes? It's a very small, although it's charged, but it's a very small molecule. So I was just wondering what limits the diffusion, because the leak that you are talking about here is through membrane transporters or something like that. So that's the question.
Yeah, that's a very good question. I would say I don't know exactly the mechanism of why the protons is not generally permeable to the inner membrane. Okay, I would say this leak, yes. So a lot of this is because of some uncoupling proteins, some extra channels. But there's people also discuss about basal leak. So basal leak, okay, I would say this is still active research. Don't know if this basal leak is still mostly because of some kind of channels, or it's actually just because the memory there's actually some proton can actually go through the memory directly. I would say, but the general, right now, the people in biochemistry is considering the inner membrane to be not that permeable to proton, but I don't know exactly the mechanism. Yeah, it's worth looking into. Yeah, thanks for the question. Okay, so great. Yeah, I like all the questions. I think this is a good sign. Okay, so for the for the last cycle, ATP, ADP. All right, so how do we measure that? All right, ATP, ADP. Okay, which is again one of the most famous molecules, right, in biochemistry, if you will, I think. How do we measure that? Okay, so this is actually a work actually in 2009. This group actually developed a biosensor, right? In this case, it's actually a protein-based biosensor, right? So again, I think this is kind of give you a different spectrum of different ways to measure, right, concentrations of different molecules. So we talk about autofluorescence. We talk about some kind of, a dye. In that case, I think you can consider that kind of a small molecule. I don't know if you can consider it really small, but it's not protein. It's small molecule dye. And this is actually a protein now. So it's a protein-based ATP sensor. It's actually a FRET sensor, right? So FRET is this mechanism of, you have two fluorophores. One is, you can consider that as a donor, right? Like kind of a energy donor, okay? Another as acceptor, right? So in this case, the donor, I think, is the CFP, and the acceptor is the M venus. So two fluorophores of different, color. And then you connect these two fluorophores with a epsilon subunit. I think this epsilon subunit is basically an ATP binding subunit. And that if ATP binds to this subunit, it changes conformation of this subunit and brings these two fluorophores closer. When they're close enough, this so-called FRET happens. Now, if you excite this CFP molecule, and then, of course, right, you excite at 435 nanometer and emit a photon at 475, but this M venus takes in this 475 photon, right, emitted from the CFP. And then, so that got excited. So energy is basically being transferred to M venus. Right, and the M venus is gonna emit a photon at the 527, right? So that basically means if you measure, let's say, either the fluorescence of this donor or the fluorescence of M venus, their intensity, right, will change depending on whether they are fretting or not, which depends on whether ATP is binds or not, and which is can be made quantitative. That this type of fretting efficiency depends on how much ATP there is in the cell. Right, so people spend a lot of time, developing those type of or optimizing this kind of biosensors such that become quantitative, right? So they also do this calibration. They can actually purify the protein outside, and then they can just add ATP. And also, they first have to show that this is specific to ATP, or like not ADP or any other things. And they did that, and they optimize the sensor that way, and then they actually titrate ATP, and then they can actually plot the ATP concentration. Sorry, the fluorescence FRET. In this case, it's the ratio between YFP and CFP, right? So the FRET ratio, and then versus the concentration of ATP. And now you got a quantitative sensor to measure ATP, which you can just express in the cell, right? So for example, here's just example, people use that. The cool thing about these sensors is that it's a protein sensor, right? So you basically, you transfect your cell. They express that, they can express that you can even target it to different compartments, right? You can add a mitochondrial targeting sequence and they're gonna go into mitochondria. Or they just stay in the cytoplasm. So you can measure cytoplasm ATP level or mitochondrial ATP level, right? There are two different pools, actually. And ATP is being transported actively, right, through these two compartments. So they might not have the same concentration, inside the mitochondria and outside. And they can do that, right, in this compartmentalized way. Here's actually, they also happen to do this hypoxic experiments. And at low oxygen, you can see this FRET ratio is significantly reduced in the mitochondrial compartments. And also you can recover that. We recover oxygen. So you can take this as dropping oxygen, you decrease ATP levels in the mitochondria. And we did that in our oocyte again, same experiment. But now I'm just showing you this ATP. And I'm looking at the CFP. I think that's the donor. I'm just looking at fluorescence. It's not the right way to look at FRET, but I'll just give you a sense that, it's fluorescence can change, right? So that means it is sensitive to this oxygen drop. And then if you really measure the FRET ratio and by the mechanism I described, you can calibrate the concentration of ATP. Okay, so one more time, it's subtle if you just look at intensity. So it's not should not be looking at this one channel and based on intensity. You either do the ratio of FRET ratio, or you can actually do what is called fluorescence lifetime imaging microscopy, which I'll be talking about in the third lecture. So, but then this measurement is telling you are decreasing ATP level when you drop oxygen. Okay, so any questions with this?
Yeah, I have a question. So
Yeah.
in the next slide, I think it's in the next slide.
In the next slide? Oh,
the one
this
with
one.
the images. Yeah. So
Yeah.
here you're saying the measurement of ATP concentration only, right? So is there a way to get the ratio of ATP to ADP like we learned in the morning? Because if it gives the absolute concentration of ATP, that's fine. But ultimately, what we would like to correlate it is probably with the ratio. So do you separately quantify ADP concentration, or there is a way to get the ratio directly by the FRET experiment?
Yeah, super good question. Super good question. I think that's what I kind of want to encourage you guys to think about. The concentration ratio, right? Even down the line, there'll be fluxes and what are the roles of this quantity. So this is a super, super good question. There is actually a biosensor just to measure ATP over ADP ratio, right? This one is called A-team. Somehow all the biosensors have funny names. This is called A-team. And the other one to measure ATP over ADP is called Percival. Okay, so this is also, I think this is developed actually by a group at the Harvard Medical School, who used to be our colleagues at Harvard, and they actually also perfected, at least I would say optimized, this biosensor, which is Percival, that happens through the competitive binding of ATP and ADP to this sensor, and that changes its fluorescence. So that it uses competitive binding of ATP and ADP, they can actually probe the ATP-ADP ratio, and that's what changes its concentration. That's what changes the readout of the sensor. So I don't think I have that in this slide, but actually we've been using that to do research. So so that exists.
Thank you.
Yeah, cool. All right, but I would put a word of caution here that all those measurements, it's easy to do if you want to just do qualitative, right? If you just want to see, okay, there's some intensity change and when you drop oxygen, it goes in the right direction, good. So that's usually easy to do now, right? With those sensors, you can just, any lab just can do that if you just use this autofluorescence, a dye, or if you can do some of the molecular biology. But the challenge is to do them quantitative, right? If I want to put actual numbers of concentration, micromolar, millimolar, millivolts here, and that's what's challenging. And I would advocate, right, the whole field based to think about how to make things more quantitative, how to do the calibration more carefully, and how to validate it across different techniques. And usually I would say even just to perfect one or optimize one technique quantitatively, it takes years of work. But I would say it's worthwhile if we can do it. And it would really greatly help address our questions quantitatively in bioenergetics. And this is basically show you a kind of, right? Okay, ambition is like you can do all those together. Oh yeah, question?
Yeah, just a short, small clarification. So in the oxygen versus ATP level decrease, the graph, can you just go back to it?
Yeah.
Yeah, so for the first 10 minutes, there's like what is going on? It's not really a linear. So are you starting the decrease in oxygen after post 10 minutes?
Yeah, I'm first not dropping anything, so it's kind of a steady state, and then I start drop.
Okay,
exactly.
Okay, and like from the very moment that you start dropping the oxygen, the ATP synthesis also decreases.
Good question. Well, I did not show that data, and I think the simple question is no, right? So this is not where the sensor is responding. There's actually a pretty large range of oxygen concentration nothing happens as far as I can measure from this sensor, for example. The sensor really responds when you are like pretty hypoxic.
Okay, because that would actually also relate with the previous thing of the loss in membrane potential. So that would relate to the ATP levels being maintained. So maybe the membrane potential that's already there is being used for that, and once it's gone, that's when the ATP levels actually start to drop.
Yeah, exactly. So I think this basically says, again, right, we want to be very quantitative in the sense that we want to measure all the ATP level as a function of oxygen. So I did not plot that plot. If I plot that, you probably would see that there's kind of plateau region and it does not decrease immediately. And I should do the same thing for the memory potential, right? I probably also should even do the memory potential versus ATP level or ATP versus memory potential. Actually, I'm doing that. I already actually have some results along those lines. And that actually is something I'm trying to use that to study quantitatively how the cycles are coupled, right? And how sensitive are different quantities to oxygen or to each other. But the simple question is like, it's non-trivial, right? It's like, even for memory potential, it does not decrease as soon as I drop oxygen. Also, NADH, no. There's actually a range of oxygen. Things are not changing, even for all the 3 cycles I can measure. And it only changes at very low oxygen. And now the question, do they change simultaneously or one is lagged behind the other? There might be some interesting stuff there, which also I'm trying to figure it out.
Okay.
Yeah. So, but actually following that, right? If you want to answer that question.
I don't,
okay.
Sorry.
Yeah.
This is related to the previous question.
Yes.
I was just wondering, do you have some kind of oxygen level control where you will like measure the oxygen over the time with the probe, but without decreasing your level? Because like the probe can act on the respiratory chain complex. And then do you think that there is a correlation time between the decreasing of oxygen because you are like doing that in your experiment and the effect of the probe that could act on it?
So what is the control you're suggesting? I missed that part.
So I was just wondering if you observe any oxygen level decreasing when you use the probe without changing by your own the level of oxygen.
Meaning without changing the level of oxygen, do I see the change of the probe signal?
No, like, because if I understood your experiment, after 10 minutes, there is a change in oxygen level because you do it experimentally, like manually, you change the oxygen level, and then it decreases.
Yeah.
But then if you are just in control condition, so you don't change the oxygen level, but you add the probe, then you should observe a little, like just a small decrease of the oxygen level, or maybe it could be a huge effect by the probe also.
Oh, okay. So you are saying that the probe change the oxygen level?
Yeah, because I know that some article try to compare what is the best between TM error or rhodamine, error 123 or something like that. And then they will say, okay, this one acts more on respiratory chain complex, this one do that. And
good question. Okay, I have to clarify a little bit that this oxygen level is not the intracellular oxygen level, it's the environmental oxygen level. That's what I can tune. So that's what I'm basically tuning. I'm flushing gas into the chamber where the oocytes are being hosted. So the intracellular oxygen level, it's actually a very good question, right? Like, did not talk about this. Like, how do you measure intracellular oxygen level? There are actually some probes people claim. If you look in the literature, there are some probes. There are dyes, right? Which is, I think, rhodamine-based dyes, which is kind of phosphorescence-based, dyes, whose fluorescence can be quenched by oxygen. So you can actually buy those dyes, I think, and then that can be used, to sort of maybe measure the intracellular oxygen level. And there's also some proteins, I think, even some phlegm-based, right? Some proteins biosensors, this type of protein-based biosensor I just talked about, that can actually also been proposed to be able to read off oxygen levels inside of the cells. Actually, also even by looking at its fluorescence lifetime change. Okay, but I have not done all those. And so I would say for me, at least, it's still a challenge to measure intracellular oxygen level. But if I can do that, I think that is something I am actually quite interested in. I can do that control, which is a good control. But the best I can do now, I think, is I can add this TMRM. I can measure oxygen consumption rate of the cell, though. So maybe that's even more direct. So I can add some level of TMRM. I just measure oxygen consumption rate, let's say at this control oxygen level, to see if that's changed or not. So that would be a good experiment. I don't think I have done that just yet, but thanks for reminding me of this. Okay. All right. So then ambition, I think this following the question. Wow. Okay. There are a lot of questions. I'm sorry. I think I think I don't have to rush through the whole thing. I guess if I cannot finish, I cannot finish, I guess.
So I was just trying to understand this data. So on this slide, does it mean that the depletion of oxygen is actually causing the increase in the next slide?
Oh.
With the data, yeah, this slide. So the decrease in oxygen, is it causing increase in NADH, increase in proton gradient, and increase in ATP? Oh, okay. All right. So, right. I have not even played the movie yet. let's play it again. Okay. All right. So I don't know if I can stop it. Okay. So there are a lot of things to look at. I think this movie is one of the most confusing movie, I think, because so many things happening. So this is I don't know if you can pick up anything just by looking at it a few times. Okay, let's first focus on NADH. This increases. Okay, so again, this is normal, hypoxic. So the intensity increases, but this is because you actually decreased respiration. Like I said, your NADH accumulated because it cannot be oxidized. So your respiration is inhibited because there's no oxygen. That's why NADH signal increases. Right, but this increase of intensity does not mean you increase activity, right? So I guess this is an important message. Is like when you see some fluorescence increase, by itself doesn't mean anything, right? It means something changed, right? It really have to know how this probe is working to know the directionality, right? So in this case, the increase of NADH under this particular perturbation is consistent with the hypothesis that decreasing oxygen lowers the oxidation of NADH, then you increase NADH level. Okay, so let's say as a hypothesis. I mean, I know it's a fact now because I can measure oxygen consumption rate, and that decreased. Okay, I go more into detail in my next lecture. Okay, so TMRM, look at this. So here you can see depolarization right here. I don't have like in a split second. Right, this is basically the steady state. Steady state. This is depolarized. Right, you see kind of become more homogeneous. All right, so that from this dye is like the dye would preferably accumulate to mitochondria than the cytoplasm if your mitochondria is very hyperpolarized. If it's not hyperpolarized, it would not preferably accumulate to mitochondria, it will be homogeneous. So this means you are depolarizing. Okay, so A team, okay, this intensity really doesn't mean much because I'm actually looking at this donor. Okay, I think I would not go into too much detail because it would be confusing. But the right way to look at A team is actually you should look at this fret ratio, right? Which gives you the fret efficiency. That tells me how much you are fretting versus how much you are not. And when you are fretting, for example, the fret efficiency increases, then you're fretting, that means you have more ATP bind, right? And if you have less ATP or less fretting, right, the fret efficiency will decrease. And then there you can actually know ATP level. This, if I were to tell you very, very, hand wavily, is ATP is decreasing, which I know because I did some calibration and also some better measurements through this fret efficiency. So it's decreasing. But then if there's an increase in proton gradient, then that's leading to decrease in ATP is...
Oh, it's a decrease of proton gradient.
Okay, I see.
Yeah, so to sum up, it's a decrease of oxygen consumption rate, decrease of proton gradient, and decrease of ATP. Yeah, I know it's a lot. That's why I think really I think even to just to get the direction right, it takes quite a few careful control experiments and calibrations. And so I guess today I would say, try to give you the tools, maybe in the tutorial sessions. I think, maybe Yash can lead a bit of a discussion about how to interpret those. I think those are, given the question, I think this is probably worthwhile to give it a shot. Okay, so, since I'm, I don't have much time anymore, but I just want to give you a little bit of flavor of, actually, I still have a little bit of even basics to cover, which is, so far I talked all about concentrations, right? But concentration is one part of the story. The other part is really the fluxes, right? If I know how much ATP there is, doesn't mean much, right? Like as somebody just asked me, maybe ATP ratio is more important because that signifies how far away you are from equilibrium. Another thing is maybe how fast you're making the ATP is actually more important, or how fast you're consuming it is more important because energy is provided on the fly in the cell, right? If you cut off this flow, then the cell is dead, basically, right? So that's why we say energy flow. That's because it's constantly turning. The wheels are turning. So these metabolic activities are characterized by metabolic fluxes, which are the turnover rate of the metabolites. So that's a very, very important quantity. So I'm sure, for example, I think Jonathan, for example, I think he will talk a lot about energy flows in terms of measuring oxygen consumption rate or heat dissipation rate. So I'm not going to go too much in details on this aspect, but I would just, for completeness, I would just say, for example, we can measure the oxygen consumption rate, which is called OCR. I think for that, I will probably leave it to Jonathan to talk about. So I probably just gave you an idea that you can measure those OCR in the bulk using commercial devices. I'm not going to even tell you what exactly this mechanism is, but you can measure OCR of a population of cells using a commercial device, or you can measure it on the level of a single cell, or in this case, maybe like a single oocyte, which is a big cell, or actually a couple of them to make the signal better. But I think I'm not going to go into too much details about those techniques. Maybe Yash can help me out maybe later, because I want to give you guys a little bit of flavor. Okay, since we have a lot of questions, I might even skip this part for now, because I want to give you a little bit of flavor of kind of a case study of which is utilizing this kind of techniques that we described. And this is work from Yash, from Needleman lab at Harvard University. And this is actually unpublished work. And I'm presenting it here because I think this gives you kind of what's the state of the art right now, right? So how do you use those techniques to actually study the spatial patterning of mitochondrial metabolism in mouse oocytes? I would not have time to do justice to this work now. I think Yash can, do it himself more in detail, but I just want to show you that we can, use those tools. Ask a very simple question. Are there any metabolic heterogeneities inside of a cell? Are all mitochondria created equal or not, right? So I'm looking at this, again, the mouse oocytes that keeps appearing in this talk, which basically, right, if you look at this cell, then now that you see a movie now, you actually can see that the spindle are right here at the cortex. You have mitochondria being labeled. They just move around, they interact. And then, okay, so now we can just use what we just introduced as this TMRM, the memory potential dye, to label or to measure, at least qualitative in this case, the mitochondrial membrane potential. And we can use sertubulin to label spindle. Now, the first question is mitochondrial membrane potential everywhere homogeneous or not? Okay, so to address that question, we basically plotted the normalized mitochondrial membrane potential from this TMRM signal as a function of the distance to the spindle. I think in this case to the surface of the spindle, right? So then this is basically the right surface of the spindle, and those are kind of the mitochondrial membrane TMRM signal. It's high, and then once you go away from the spindle, like into this direction, the membrane potential decreases. And this is averaged over 29 cells, so it's a very significant reproducible pattern. So in short, mitochondrial membrane potential is not uniform, and it's heterogeneous, and it decreases away, in this case, it decreases because we know the membrane potential is high when the TMRM signal is higher, as you can see from the oxygen drop experiment. that actually shows this relation. So therefore, you have higher membrane potential here, close to spindle, lower away from it. Okay, so then we are thinking about possible mechanisms. So I probably, okay, since this is not a research talk per se, I might even just see if there's any questions here. I don't have to go through all the details, but I want to see if this motivates people to think about things. Or people want to hear a little bit more about what we did. Yeah, go ahead.
So maybe given the time, I think we can just open to discussion, right?
Yeah, maybe let's open to discussion at this point.
Yeah.
And if you want to know more about this, I would say Yash can definitely tell you more about this.
Okay, so let's thank Xingbo for the very nice lecture.
Yeah.
Questions?
Questions, yes.
Yeah, so the heterogeneity in membrane potential that is seen here, has it been correlated to like other measures that you discussed before, and like let's say the ATP probe, because like the spindle formation and everything will require a lot of ATP. So is that also heterogeneous?
Very, very good question. Okay, yeah. So for ATP probe, yes, we did do the measurements. We have not quantified, let's say, okay, because the ATP probe does take a long time to have it work. Imagining expressing proteins in the cell cycle arrested oocytes, not easy. And after many years, we finally got it to work more or less. So we actually do have some preliminary data about the ATP concentrations in the oocytes, but we did not have the chance to quantify that around a spindle or not, around a spindle just yet. So therefore, I don't have an exact answer for that. So I would say it's a progress, it's work in progress. But we did look at NADH. Okay, we look at NADH autofluorescence, and that also has a gradient consistent with this.
Okay, and yeah, that has a similar pattern.
It also has a gradient, of course, in terms of what do you mean by similar? It's like also the same kind of in terms of intensity or not. I think, like I said, intensity alone does not mean much, right? But they have the same length scale. Let's put it that way, right? I think that could be safe to say. Yes, no? Yeah, good. All right. Any more questions?
So along with the localization of mitochondria in that region, have you also thought about, or maybe already looked into, if the glycolytic enzymes are also phase separated nearby to those mitochondria? Because ultimately, to run all that mitochondrial respiration, you would need the glycolytic. Yeah.
Oh, this is a very, very good question. Yeah, I think we did not look much into glycolysis. Okay, because for oocytes, right, the current research shows that glycolysis is not happening much at this stage. It's dominated by mitochondrial respiration because first, you don't even have much of a GLUT transporter at this stage. So some people even say you're not actually, glycolysis is almost not happening at this stage. The other side of the story is actually there are some supporting cells called cumulus cells that actually around a spindle, which is not here. They are being denuded in this case. But when they are like in vivo, when they're like maturing together, when the oocytes are maturing, it's matured together with the supporting cells, and the supporting cells are doing glycolysis and make pyruvate, and pyruvate being transported directly into the oocyte, and they just start from pyruvate, and then the mitochondria taking pyruvate and do respiration. So therefore, I'm not sure how much relevant is glycolysis in this case, but what you said is a very general question. From a general question perspective, I think it's a very interesting idea. I wonder also, do you have any hypotheses or speculations, if I may ask?
In general, like it's seen in bacterial pathogenesis, and also if some pathogens will rewire mitochondrial metabolism and they'll make metabolones, which are phase-separated glycolytic enzymes, which are present near the mitochondria. So I was thinking in that context, if that is something that also happens here. But what you explains that. Thanks.
Yeah. I think that, some of my colleagues here in Dresden are working on those. I think I'll just stay tuned. Hope there will be more progress along those lines. So,
I also
okay, go
also have a
ahead.
question.
Yeah.
So, for example, in some other cell types, they will also try to observe the spatial patterning of mitochondria, and they will observe that there is more density at the periphery than the nucleus, for example. Then I was wondering, my first question was in the oocytes, like where will be the nucleus? And also, do you observe like an increase in functionality and density near to the spindle, or just the functionality? Like, is there any mobility, do you think, about the mitochondria?
Oh, wow. Super, super good question. I would say, actually, I meant to go into that through Yasha's project, but unfortunately, don't have time to answer that very quickly. Okay, here, we don't have a nucleus because nucleus already disassembled, right? And then you are at metaphase. If you wind back time, you would see at GV stage, there's still a nucleus, and they break down after like about 15 hours, you are here. So there, actually, we're also, in my group, we're trying to study that. We're trying to study if there's any patterns of metabolism around the nucleus and away from it. Maybe in my next lecture, I will give you a little bit of flavor of some type of radial patterning of mitochondria, right? But still at this stage, so still no nucleus. But so under that kind of maturation of oocytes, I think that kind of nucleus and away from nucleus is probably a relevant question. My mobility, actually, I would say, Yasha can tell you much more about it, and you can see that the mitochondria are moving, right? So here, they're just moving, actually, like that. And actually, during maturation, they move even more, right? And actually, there's even work showing that the movement of mitochondria and actually the movement of spindle in terms of making the spindle go towards the cortex and maintain it there, mitochondria is playing a role. And also, in this case, we are actually asking a very big question, right? Like, what is the mechanism for this kind of patterning? Is the mechanism due to some type of signaling, right? I mean, signaling hypothesis, for example, same mitochondria are respond to some kind of signaling gradient, or here is the self-organization hypothesis, which functionally distinct mitochondria have different motility, like so. So the mitochondria are different, they just move, differently based on their metabolism. I think which is a very bold hypothesis, but I think we are having some evidence to show that this is not completely out of the question. So therefore, just stay tuned, and hope Yash can tell you guys more about this. So I think, okay, I guess I'm running out of time, so maybe I'll just wrap up. But I would again say, okay, yeah, as you can see, this is a very cool project from Yash. So talk to him. But he will lead again the TA session, which he will be talking about the reaction diffusion modeling of ATP and ADP in the cells. So then you guys can actually think about how to incorporate some of those techniques we discussed in the lecture. What things you can measure now, how do you use those measurements? How do you put that into, for example, a framework of reaction diffusion to help you understand the patterning of energy flows and maybe even energetic costs of cellular processes. So I would say, Yash, will help you guys with that.
Thanks. Is there any more questions? Otherwise, we can thank Xingbo again. Nice lecture.
Okay.
And
thank
I
you very
remind
much.
you, we reconvene at 4 for the TA session. Thanks.
Okay. All right. Thank you.
We have coffee. This is